our speaker, who is one of my newest colleagues. Lydia Kisley grew up just down the road in Mentor, and after studying at Wittenberg University for her bachelor's degree, went off to Rice in Houston to do her PhD in physical chemistry. She did a postdoc at the University of Illinois at Urbana-Champaign, where she was named one of the Forbes 30 under 30 healthcare list of change makers and innovators. We were so pleased that she was able to join us on the faculty in January 2019. So she's been here just over a year, back around the corner from her hometown, doing really exciting work in microscopy. And she's going to tell us all about it tonight with lots of great pictures, I'm told. Dr. Kisley, why don't you take it away?
Yeah, thanks for the introduction, Glenn. Let me share my screen. And like it was noted when I was teaching this semester, using the chat box is the best way to get my attention if you guys have questions. And I'm happy if anyone wants to interrupt and ask any questions throughout the talk. And
but I'm going to mute everyone just so it's easier for you.
Okay.
Let's see. Hopefully, I will just unmute you though. Okay.
Unmuted. You guys can hear me.
If people have questions during the talk, they should type them, not ask them.
Right.
Okay. That sounds good. So I'm a microscopist by training. I think it's an awesome technique. And it's a way for me as a physical scientist to visualize how nature works around me in the same way I was trained to, thinking about molecules, thinking about atoms, and thinking at these small length scales. So I'm going to be talking about and show you guys what some of the cutting edge or current methods are to look at the world around us with different forms of microscopy, and also talk about how we've gotten here and what some of the history of microscopy has been, and also the recent advancements that have led to these methods. And of course, since we're doing this remotely and over Zoom and stuff, I've tried to look at some applications that might, or some examples that are related to our concerns today with the coronavirus, our immune systems, and the like. So I know with this being a talk through the Institute of the Science of Origins, there's very large length scales or range of length scales that talks are over in this series. So to give you guys some perspective of what length scales and what distances I'm interested in with microscopy, and also time scales as well, I have this scale bar here of, I know there's talks about the universe, and we see that with the backgrounds of some of the people watching, and these are all in units of meters, where this unit here with the 10 to the power 28, that means 28 zeros behind that 10 there. And I know there's also talks about anthropology, about mammals and the like, and that would be on the order of 1 meter here, 3 feet or so. And then I know there's talks on particle physics and getting down to really small length scales. But the area that microscopy covers is distances that we can't really see by just below the naked eye and getting down to the molecular level. So we see different organisms here, but microscopy in different forms really spans from, I would say, hundreds of micrometers or millimeters. A good point of reference for what 100 micrometers is, and kind of the limit of your naked eye resolution would be the diameter of a hair, which is 100 micrometers. And getting below that, the cells in our body are 1 order of magnitude less, so 1/10 of that size. And then the virus that we're all concerned about is 1/1000th of the width of your hair, 100 nanometers. And then a lot of the proteins or therapeutics that they're testing for drugs would be 1 nanometer, or proteins, if we're looking at biological treatments, would be 10 nanometers. So this is kind of the scale that we're looking at with different forms of microscopy, getting down to individual cells, individual molecules, individual macromolecule structures as well. So that's a point of reference of the scales we're interested in. But when I bring up microscopy, probably the first type of microscopy, or what people think of with microscopy, is thinking of looking a light microscope, like this very simple picture over here of a compound light microscope. And what a light microscope is a fancy way of focusing light and a fancy combination of lenses. It's not just a single lens like a magnifying glass, and I'll talk about that later, but a way to control multiple lenses together. Where all light microscopes will have a light source. So in this very simple one here, it would be light from the room reflecting off this mirror and being focused onto the sample. That light's being delivered to the sample. It could also pass through an objective to be tightly focused, and then imaging that sample, and then it passes through an objective and different lenses here to then be detected, where a very simple light microscope like this, where the detector would be your eye. Or for other types of microscopes, you can put different types of cameras there. So it's a way to control the direction of light and delivery of light to a sample, and also detecting that light to form images. So move this guy over here. Also highlight that microscopy, different forms of it have won different Nobel Prizes throughout the years. It's been a regular occurrence in different awards in chemistry and in physics. The video I have shown here is from one of the Nobel Prize winners in 2014, Eric Betzig, who is now at UC Berkeley, with some of his high power light microscopy to look at living organisms, living cells with light. And that's really the power of light microscopy, is you can look at dynamics at fast time scales. So in this video I'm going to show here is looking at in real-time imaging dynamics within a zebrafish embryo. So in this image here, these blue droplets are some foreign polymers that were introduced to the cells, and then these guys running around are actually immune cells collecting this foreign object within the zebrafish embryo, and you can see them moving around and doing their job in the immune system of the zebrafish. In this angle here, this will be looking at real-time of the zebrafish as well, where there's a blood vessel over here. You can see cells passing through. You can also see cells moving around, that there's an immune cell, and they'll freeze, showing that there's pressure relief valves, cell division taking place that they can image in real time. And then you can see here with this last part that they can track the individual immune cells diffusing around, or moving around, and they can look at the speed, where here is the trajectory pulled out from that movie, where the different colors represent how fast that cell was moving around. So I mean, this video is just a very beautiful example of how you can see life taking place in real time with a light microscopy. But a limitation with light is the resolution or the wavelength that you're using with light. So this will limit you to hundreds of nanometers of what you can image, unless you use some tricks that I'll talk about later. But a way to reach even higher resolutions is using electrons instead of photons or light. And as an example, electrons can have a wavelength that's much, much smaller. They can be focused much tighter. And this is just a very relative scale of just showing you what you can resolve will be based on the distance between these peaks, how tight they are. So when scientists were running into limitations of what they could resolve with light, as you remember, the size of viruses or proteins are below 100, those hundreds of nanometers with light, they turned to electrons instead. And you can see that this won multiple Nobel Prizes in physics, chemistry, and even most recently how it's been applied to looking at biology with electrons. The 2017 Nobel Prize in chemistry went to cryo-electron microscopy. And I have an example image shown here of what cryo-electron microscopy images look like. And this is from a very recent paper that was published about 3 weeks ago, where they were looking at the spike proteins that are on the COVID-19 virus capsid. So if you think about that cartoon that's in all over the media, where it's a gray, I guess, spherical particle, and they have red spikes coming out of it, that's what they're looking at here. And in this cryo-electron microscopy image, you can see that there's areas where it's like darker gray, and those are actually the spike proteins, many of them, hundreds of them, that they flash froze, so the structure was maintained. So this image may not look as interesting, but what they do is they take all of the different images. Let's see with the chat. Okay. No, so the question is if this is AFM. No, this is with electron microscopy. So I'll talk about AFM on the next slide, and I'll define what AFM is then. So here they take all these patterns that you see. These are all individual spike proteins, and by collecting many, many images of those, they can then produce a very high resolution image of what the spike protein looks like. So you can see here, this is what's put together by looking at the patterns and the images of all hundreds, thousands of those proteins. And you can see that this is what the spike protein looks like. You can see this local resolution tells you how confident they are and what spatial dimensions that they have. So you can see that this is listed in angstroms, which is another unit that means 1/10 of a nanometer. So we're at atomic resolutions here with the structure of this spike protein in the coronavirus. And you can see that they were able to image this protein under 2 different conditions where they saw that there's an opening in the spike protein, that they believe that this structural change here might be important in the spike protein landing on the receptors on the cell surfaces in our bodies that helps the virus inject the nucleic acids and cause the infection. So by identifying that there's a structural change here, and that's the power of electron microscopy, this is a targeted area where they might be doing modeling with small molecules or drugs to try to inhibit the recognition of these spike proteins with the coronavirus. Yeah, so the question here is, why is it colored multiple colors? So this is telling how high of a resolution, how much confidence they have in the structure here. So that's what's leading to the different colors. So you can see that it's red in this open area because there's probably, if that is undergoing some dynamics or having some different amounts of opening, that would lead to variation there. So that variation would then lead to higher uncertainty in their resolution. So that's what the color is representing, how confident they are that this atom is, or this portion of the protein is actually there versus not. So that's with electron microscopy. And then related to the other question about AFM, that's what I'm talking about here, which is using force or dragging a tip around to be able to look at the structure. So instead of collecting an image over a wide area, what scanning tunneling microscopy, that's what this cartoon is showing here, or atomic force microscopy, which is shown here, is doing is, if we take this tip, you can see here, a very high ultra fine tip, and here they even put a carbon monoxide molecule on the tip. And they scan it over a surface that, by looking at how this tip deflects or changes based on what's on the surface, they're able to produce based on actually touch, looking at electrostatic interactions, or looking at electrons tunneling, that's the scanning tunneling microscopy. And this method won the Nobel Prize in Physics in 1986, but it's reached very high resolutions, where here I'm showing this is a molecule that's undergoing a reaction, where this is an organic chemistry representation of a molecule, where each of these vertices here would represent a carbon atom. And you can see that with very high resolution, once again, this is 0.3 nanometer resolution, they can actually image the shape of this molecule, and they can see it undergoing reactions and forming a different structure. So this is amazing, I think, based on like when I took organic chemistry, like you draw these sketches, but you have to, I guess, it's all on a chalkboard, that here, this is actually an experimental way of visualizing reactions taking place, showing the power of how high resolution microscopy has achieved now. This is actually a carbon atom, or the electron distribution of a carbon atom mapped out here. So that's to give you guys an overview of different ways that microscopy is performed, either using photons and using light microscopy, the most traditional way and most historic way of microscopy, or once the limits of those microscopy were limiting scientists at what they were looking at, using electrons or using force, ultra fine tips to look at, achieve very high resolutions. But with all this perspective of, with all this cutting edge microscopy, question is, how did we get here? And I always like learning about the history behind the science that takes place. And if you guys, I know some of the people in the audience are members of the Case campus, but also people in the Cleveland community, you can see a great exhibit, I think it's a permanent exhibit, of some of the history of microscopy right on Case's campus at the Dittrich Museum of Medical History. So that's a medical school library. So it's right across the street from Severance Hall at the corner of Adelbert and Euclid. And I think the renovations might be done there, but if it's open up, they have very nice displays of old microscopy setups and some of the history there. This is some of the examples of the microscopes they have. These are mostly dating from the 1800s, and I'll show some specific examples a little bit later. But to start off with the history of light microscopy, that's what I'm going to be focusing on, is the most simple microscope you can think of is a magnifying glass or a single lens, which, I mean, there's long history of that being used or reported. Maybe the anthropologist could comment on that, I think in archaeological digs, maybe. And there's also reports of people writing about even just using a water droplet to magnify an image. So that was the limit of magnifying images up until the 1600s, was using a very simple single lens. And in the 1600s is when the first compound microscopes were developed. So that would be, instead of using a single lens, which then flips the image, using two lenses to improve your magnification and also get a picture of the real image of your sample. So that was developed by an unknown scientist in Holland in the early 1600s. There's not a good record or any scientific publications of who developed that first compound microscope. But one of the scientists who first used a compound microscope or recorded and reported scientific results was Robert Hooke, an English scientist. And in 1665, one of the founding microscopy reports was his report, Micrographia. And this is an example of his compound microscope, where there's a lens at the bottom, the sample would be down here, and a lens at the top. So there's multiple lenses present. You can see over here, this was actually a container of oil. So he would have an oil lamp, and that oil lamp would then serve as a light source that would then be focused through this blown glass bubble down to the sample over here. And Robert Hooke made the important, I guess, nomenclature of naming the cell based on his observations that he reported in Micrographia. This is the image he sketched by hand. That's something you have to think about historically before photography was developed, that all these observations had to be sketched by hand. And this is his observation of looking at cork under his microscope, where he saw this pattern of structures that he named cells because they reminded him of the monks' sleeping quarters. And that's why he named it a cell structure. So he made that observation, but he made no connection to the cell being the fundamental, I guess, unit of biology. It was more of just his description of what the structure looked like. So while in biology, everybody talks about cells, and that's a key aspect there, and Robert Hooke gave that name, it is interesting that he didn't really relate it to the biology of cork being biologically derived from a plant. So with compound microscopes, people were able to achieve some good imaging, but the problem is with these compound, having multiple lenses and being able to polish and make high quality lenses was difficult, and it was kind of stagnant with the capabilities of compound microscopes in the 1600s. There was another advancement by Antony van Leeuwenhoek, also in Holland, in the later 1600s. He was able to make amazing single lenses and polish amazing very small lenses, and he could get a very high resolution. So this is an example of one of van Leeuwenhoek's microscopes where he would polish a very tiny lens. This is probably only a couple millimeters here, and place it in this device. So it would be in this small, tiny hole here. He would then place a sample on this pin needle over here, and then he would have to hold it very close to his eye, and looking through one eye would be able to look through this very high quality single lens microscope. But he made some of the first, his great capabilities of making these very fancy single lenses outperformed the ability of compound microscopes. So he made some of the first observations of, he looked at his saliva and was able to see bacteria in his saliva. He was looking at pond water and he could see flagellum on bacteria, and he made many of the first observations of those microscale and microorganisms. And he gave the name to them, animalcules, and he was able to report his observations there. And I have this, I guess, old picture of showing someone looking at, I guess, these animalcule creatures, and that actually became, I guess, later in the 1800s when microscopy became more accessible, also somewhat more affordable, that in the 1800s, this actually was a form of entertainment for socialites when they were having parties. And there's a great example in the District Museum of a kit like that, where people, when they were having people over, they'd say, hey, let's go grab some water from the pond. Let's look under my microscope. I have this very fancy microscope. And that was a form of entertainment of looking at these microorganisms. Something to note with Anthony van Leeuwenhoek is that many of his lens polishing practices, he kept a secret. So while he was able to make some really cool observations, they kind of went with him. And also, even as recent as like papers from, I guess, like the 1960s, people were trying to reproduce how he could have polished and made these very fancy small lenses. So his lack of dissemination of his methods kind of held back other scientists from being able to look at biology and life at very high resolutions. So between, I guess, the 1600s and not until the 1800s, people, it was kind of stagnant in either using not so great compound microscopes, and I guess whatever Van Leeuwenhoek saw. Even in the 1800s, Robert Brown, a Scottish scientist, was still using, I guess he was using a single lens microscope, and he was able to make observations in observing cell nuclei on onion roots. I know I did that in high school, taking some images of that. But also, so that would be in biology, he made an impact with what he was observing. He was a botanist, but also in physics, and he made some important observations. And Robert Brown used his microscope to look at the movement of pollen grains, and he saw, which he defined as, it was named after him, Brownian motion. So in this video here, you can see pollen grains jiggling around and diffusing. And Robert Brown was very excited by this, and he thought he found the essence of life based on this movement around. But then when he took a look at, I guess, grains of sand or dirt, he also saw that they were jiggling around. And this is defined as Brownian motion, or random movement of particles based on, I guess, thermal fluctuations in the environment, which is a whole area of study in the physics area. But microscopy was key for that. But it wasn't until, I guess, the late 1800s that microscopy, light microscopy, reached its limit for a while. So in the 1870s, Ernst Abbe in Germany, and also his colleague Carl Zeiss, were able to improve the lenses and optics that they were able to develop. And they were able to create great microscopes that reached the fundamental limit of light, which Ernst Abbe defined, which I have written out here. And this resolution is the wavelength of light divided by approximately 2. So with visible light, if you're looking at green light, that would be 500 nanometers or so. So thinking back to that scale bar on the second slide. So for light microscopy, this is around 250 nanometers. And what this means is that if you're looking at something, let's say a single protein that's 2 nanometers, or let's say a virus particle that's 20 nanometers, or something larger like that's 200 nanometers. If it's below this limit, you're always going to see this diffraction limited pattern here. So they can be vastly different sizes, orders of magnitude and difference in size, but you'll always see this blurred out spot based on this limit of light. So this was established in the 1870s. And then where this limit also becomes a problem, not only just understanding the scale there, but also if you're looking at multiple things at once. So you can see here, if I have two things that are close together, less than this diffraction limit of light, you start to see their patterns run together. So you can think if you're trying to look at something in a cell environment, let's say you have millions of molecules that are labeled or have these emission patterns, you're just gonna have a very blurred out image. So it becomes a big problem. So this was long established since the 1870s, and this diffraction limit of light was thought to be true up until the 1990s. And I have here a zoomed in image from the Dietrich Museum of some of Ernst Abbe's microscopes. So he actually helped start with Carl Zeiss Microscopy, which is still one of the leading microscopy companies today and instrument companies today. And so these are some of the example microscopes you can see down here at the bottom, 1880 to 1890, 1873. So I think it's very cool that they have some of his microscopes that when he was alive, when he was running this company, that we have examples here in Cleveland to take a look at. So this limitation, this diffraction limit of light that creates these blurry images, long established, and that's why when I was talking about as a start of the presentation, that's why electron microscopy got developed, atomic force microscopy got developed. We couldn't overcome this diffraction limit of light. We couldn't overcome it. So people turned to other interactions between particles or forces and matter. But in the 1990s and 2000s, theory, it was first established by theory, and then experimentalists started to came up with ideas and came up with tools to be able to overcome the limit, this diffraction limit of light. And the idea behind overcoming the diffraction limit of light is that we can tell where the center of a peak is or a diffraction limited pattern is compared at higher confidence than the entire emission or that entire circle. So I guess 2 slides back here, I'm just showing this in 2 dimensions where the color is representing the intensity. This image here is more of a 3D view of that pattern. And you can see that there's a peak there. So instead of describing, I guess I have this image of this mountain here to give an example that when we describe a mountain, we typically describe it by its summit, where that location is as opposed to the entire base. And so if we describe our microscopy images by the summits or the peaks, that center, we can overcome that diffraction limit. That's the actual entire, I guess, area that the mountain covers. So I have, and this is called super resolution microscopy, where it means you're beating that diffraction limit of light. That is, it's also, people say nanoscopy because you get down to tens of nanometers. So the way to beat this diffraction limit is also key that you only have one molecule on at a time. So I have a cartoon here showing, let's say we have 3 things that we're looking at that are separated by less than that diffraction limit. If we take a look at them all at the same time, we have our blurred out spot. But if we only have one of those on at a time, one of those emitters or a single molecule, we can tell where the center location is. So we can fit this, we can find where that location is, and we can map where that summit, that peak is. We can then have that molecule turn off. We can use special chemistry, special physics, photophysics to be able to do that and turn a different molecule on. And we can do this over and over again, keep finding those peaks in those locations, and we can build up an image. Instead of having this blurred out image, we then have a map where we resolve where those actual emitters are. So we can get down to tens of nanometers as opposed to hundreds of nanometers. And the fact that this is a light microscopy, that means we can look at living things and is compatible with biological environments. So the theory was developed by Stefan Hull in the 1990s, and then the experimental demonstrations were done by 2 separate groups in 2006 with papers that were released a month apart or so. But I think it's a creative and great way to show that rules that were long established since the 1870s could be broken. What is the color? Okay, I'm sorry if I missed the question from 747. We can ask about that later. And yeah, there is the question about, does this introduce a lot of uncertainty in systems where you're not aware of all the elements? Yeah, so you have to, many times, the things that are turning on and off are, you know exactly what those are targeting. So you might label just the membrane of a cell or just a certain structure. And then also it is going to limit being able to have these molecules turn on and off and localize where they are. That's at one time scale. And then any dynamics that you're trying to look at would have to be at a somewhat slower time scale. So many times this technique is used to look at fixed samples sometimes, but people do look at dynamics. But we can talk about later with the questions about what those time scales are, what those limitations are. Okay. So with these capabilities and with the ability to get super resolutions, let's see, it's frozen. Okay. Sorry. So as an example, I guess on more of a macro scale of how this technique works, if any of you guys, I don't know if Professor Rosenblatt, Chuck Rosenblatt from the physics department, he should have been in Paris right now with a group of physics students talking about science in France. But if they were there, they might have been able to see the Eiffel Tower, where at night they'll have their light show on the hour and all these lights fluctuate, and someone put together localizing where those lights that are turning on and off. Let me try to replay this. By localizing where these lights are turning on and off, they can find the center, and then you can see there's a much higher resolution of the Eiffel Tower here. I don't know why the image is somewhat cut off, but you can see here, this is on a much larger scale than what we're doing on our microscopes, about 100 meters. But you can see if you took, let's say you average all those lights together, it would be more of a blurred out image compared to this reconstruction, which I think is a nice, acute demonstration of how super resolution imaging is working on a macro scale. So I mentioned the strength of getting down to these tens of nanometers and its compatibility with living environments. Getting down to that 10 nanometer scale was so key of looking at inside cells. So this is 10 microns is typical diameter that you can approximate for a cell. So things would be, if I'm saying 10 nanometers, that's 10 to the third, or 1,000 times smaller than the width of the cell. So things within the cell, I have this little red boxed out area. People are looking at structures within the cell. It's at that 10 nanometer length scale or single nanometer length scale. So people are able to achieve that. So you can see here, this is what that blurred out image would look like. This is along the axon of a neuron. And then this is the super resolution imaging, where you can see some striations and striping here. The color is representing the depth, or the Z plane here, but they were able to look at how neurons, let's say the neuron, the longest neuron in your body goes from the base of your spine to the tip of your toe. How does it maintain that structure and not collapse on itself while you're moving around? Well, they saw that this neuron has ring-like structure that helps maintain, I guess, the stability and the structure of that neuron along long distances. So that's with a static cell, but also looking at dynamics, people have looked at how molecules move within cells. So this is a myosin protein walking along a filament that's able to do transport, active transport within cellular environments. So that's a dynamic process. And then in terms of materials, people have looked at catalysis, so how fast reactions take place on nanomaterials. So this is looking at a gold nanorod that would be used, let's say, in the petrochemical industry to try to catalyze our crude oil to something we can use for in our cars or for plastics. They were able to map where reactions were taking place. And since these particles are so small, you can see that the length scale here, they were able to map below that diffraction limit of light, where it is higher in the center. And And then this is a hard material, but people have also looked at soft materials. At Case, we have a lot of polymer scientists, so people have been able to target and map polymers that have two different blocks to them, two different materials. So you can see here, red targets one type of polymer in the block and green targets another, and they're able to map that below the diffraction limit. Again, here's the scale bar of 1 micron. You can see across this polymer would be below the diffraction limit of light, so they can get structural information or chemical composition information there. So the power of light microscopy, and especially being able to look at these small length scales and also faster time scales, these time scales are milliseconds, is very powerful technique and is what I'm using in my own research group. And the gap that my research group is trying to fill is that I'm very inspired by how all of the ways on the previous slide people have used super resolution imaging in the biophysics area, where people have taken cells and looked at them in the complexity that they are. Cells are very complex environments with millions of different molecules. And then on the flip side, the materials community, how I just mentioned those nanoparticles or for catalysis or polymers, it's a much smaller community that people have been looking at this super resolution scale, but it's mostly physical scientists where they say, I want a really well-defined system. I want to know that it's just water, it's just some type of molecule and one type of nanoparticle. It's like 3 things. So it's very simple, but it's very far removed from the actual application. So I'm inspired by the biophysicists, but I want to look at some material problems with biomaterials and soft materials. And can we move the super resolution field? Can we use this microscopy to look at new problems and complex conditions, very motivated and relevant to problems in society, problems in medicine, problems in industry? So some of the projects that I have going on in my group include looking at corrosion at these very small length scales, very fast time scales. So can we use that turning on and off of different dyes to be able to detect when corrosion is taking place? Can we assess anti-corrosive materials? And can we understand how corrosion is taking place? Can we detect it faster? And can we assess materials very quickly? So this is an example of, since we're working remotely, we haven't been able to do as much work in the lab environment. So this is actually a simulation that looks very similar to what we're expecting in some of our preliminary work on corrosion, where these gray sites are where corrosion's taking place. The blue are molecules diffusing around. And then these bright spots are what we would localize, or a turn-on event telling us corrosion is taking place. So at the end, we can have a map at very small length scales, tens of nanometers, right when corrosion is starting and taking place, of where corrosion is taking place and detecting it much sooner. I'm also very interested in how molecules move around in porous materials. And this is very motivated by how porous materials, let's say like, can be used to separate molecules. So this is used in the pharmaceutical industry, also petrochemical industry. Can we look at how we can optimize separating things based on different properties? Here I'm talking about chirality, that has to do with the geometry of molecules. But can we image that at new length scales? And being able to optimize separations is a very expensive process in developing new drugs. It takes a lot of time, and it's really driven by trial and error approaches. So can we understand the physics behind what causes different separations? And then finally, on the most biology side and medical side, I'm interested in how proteins move outside the cell and how signaling between cells by proteins changes with disease. So we're able to look in our microscope with things, let's see if this video plays. Fortunately, the video is not playing, but we can actually track around similar proteins diffusing around. We can map where they're able to access in porous environments that's outside of cells, and how fast they're moving, what their stability is. And what we would like to look at is, as disease progresses, here I've listed specifically a signaling protein related to muscular dystrophy, where the pathology, the disease of muscular dystrophy causes stiff tissue, which means from the protein's perspective that there's more material, that there's more confinement, there's different chemistry. Can we map that at these very small length scales, where from the perspective of the protein. So I love that microscopy gives me a tool to visualize these small length scales and that I can apply it to very diverse and interesting problems with this great tool. So that's most of what I had. I thank you guys for coming remotely and for your attention. I'd be happy to answer any questions.
Great. Girl. Oh, I'm going to mute everybody again except for, Dr. Kisley. And if there are any questions, go ahead and unmute yourself and ask.
This is Mark Given. This question is sort of an aside, but I wonder if you have any indications of when college faculty members like yourself will be allowed to go back into their labs to continue their research.
Yeah, I mean, you can ask department by department. I mean, some people are still going in the lab, some people aren't. That's definitely an active point of discussion between many scientists, both at CASE and all over. So it's all based on, I guess, trying to have some distance practices, the safety, the availability of PPE, because you can even talk about, okay, we donated a bunch of gloves to the healthcare workers, but we need to make sure we can also have those if we're doing certain types of science as well. So I think just like everything right now, everybody is trying to find that balance and hoping to go in sooner versus later, but a lot of individual and group decisions are being made.
Great. Other questions?
I have a question about the use of light to resolve items smaller than its wavelength, particularly if you can resolve something at the atomic scale. At what point do we reach a limit imposed by the uncertainty principle?
Yeah, so I would say that's very true. You have to consider the uncertainty principle. And I think when I was talking about scanning tunneling microscopy and atomic force microscopy, that's where you start getting down to that atomic scale of the limits. And also, I guess in terms of light microscopy, you have to think about people are able to detect single photons, and with that detection, there's uncertainty taking place. So I'd say with the microscopy I'm working with, we're not encountering the uncertainty principle that frequently, but once you get down to that single photon or those electron distributions, that is something that you have to consider.
Anyone else?
This is Kingman.
Great.
Your muscular dystrophy example was very intriguing because it showed how molecules outside the cell. But I'm also thinking about the issue of how neural transmission occurs and how the idea of having a certain region of the cell attached to to extrude things that go into another cleft and then get picked up and then disposed of. And are you able to get a handle on those sorts of dynamic processes that might happen with those sorts of disorders?
Yeah, so that's a great question, because right now I'm working on writing a white paper about imaging the extracellular space between neurons. That a lot of, I guess, neural studies, people focus on the electrochemistry or the structure or what's taking place at the membrane of the neurons or within the neurons. So right now I'm writing about or proposing, can we image that extracellular space between the neurons and the glial cells. So I would say in between the clefts of neurons, that would have to, I know the processes of ion transport between neurons is very fast. I think with the type of microscopy I'm using, it would be too fast. And some other forms of light microscopy might be able to get down to like microseconds. Yeah, I'm also intrigued by what's taking place outside of cells in the brain as well. Because I think a lot of biochemistry and biophysics is focused the cell, the cell, the cell. There's this whole area outside the cell that also has to play a role somehow.
So if you step back from that though and say, I think that hydration in particular areas is also important for catalytic, numbers of molecules of water, but that just means distance and water and swelling. And for instance, during wakefulness, apparently the cells are bigger and the spaces are smaller than during sleep, when the spaces are between are bigger and the cells are smaller. And maybe those time scales are able to be picked up. That's
and
I was also thinking.
Yeah, and I've seen some work where people have done another type of light microscopy where they've looked at that extracellular space and they put it in, what you're saying is the amount of water. They've been able to take it in salty solutions versus more aqueous solutions and seen those changes. And that was over, I think, the course of minutes. Yeah, but so yeah, your guess is completely right that people have looked at that.
So you're talking about new applications of these techniques. Are there also new techniques in super diffraction microscopy that are coming, that you foresee or that are coming out?
Yeah, I mean, that's definitely a whole area of light microscopy is people focused on the hardware side of things, and also the software. So I'd say in my group, we'll come up with new software techniques, not as much the hardware, maybe a little bit, but people are coming up with new ways to, a big question is looking at information in 3 dimensions. So many times with these light microscopies, you have to take a slice, then you have to wait, you have to take another slice, another slice. How can you get that 3D information all at once? So people are developing hardware approaches with that. And with that can require, different lenses, different objectives, very creative ways. But I would say in this area of this type of microscopy, people will work on hardware side of things. There's also a lot of time spent in front of the computer analyzing these images, and then also looking at, I guess, the samples. So depending on the microscopist or the lab you're working at, you'll have different mixtures of those.
Great. I think we have time for maybe one more question.
Not really.
Not really. Oh, not really. So you can probably email a question to Dr. Kisley if you'd like. What's your email? It's
here. I can share the screen again if that's fine.
While she's doing that, I'll remind you about Flat Earth on Saturday at 7:30. Same URL. Here we go. Lydia.Kisley@case.edu.
great.
Thanks, everyone.
Thank you so much, Dr. Kisley, and thank you everyone for coming and joining us. And we look forward to seeing you Saturday at the